Hello all, we are the Yellow Yak Private Investigation Agency. Today, we will be presenting a case about plant and fungi symbiosis, and how an ectomycorrhizal nitrogen economy facilitates monodominance in a neotropical forest. This neotropical forest will be found in Central America, specifically in a country named Panama. Let's take a look. Here, we have a montane tropical forest where Oreomunia mexicana trees make up more than 60% of the forest canopy, and an excellent example of monodominance. But why does this happen? One theory points to plant-fungi symbiosis. About 440 million years ago, plants transitioned to land from the sea. With them came fungi, specifically mycorrhizal fungi. This class of fungi are recognized for their symbiotic relationship with plants. Through the thin branching hyphae filaments of their mycelia, fungi are able to access nutrients deep in the soil that otherwise cannot be reached by thick plant roots. To compensate, plants associate with these fungi in order to obtain nutrients, providing the fungi with sugars and water in exchange. Thus, the fungi and plants transition to land together, exhibiting a mutualistic and symbiotic relationship that still exists today, and the effects of which are still well observed, most notably with the arbuscular mycorrhizal fungi, or AMF. These make contact with the plasma membrane of root cells. However, associations can also occur with ectomycorrhizal fungi, or EM fungi, which function by forming a sheath around the root tip of plants and penetrating the root structure between root cells. In previous work, it was found that EM fungi only contribute to about 6% of neotropical plant-fungi associations when compared to AM fungi. However, they accounted for nearly half of the associations in monodominant communities compared to AM. Thus, the investigation focuses on EM fungi and how their interactions with the monodominant community of Oreomunia trees in Panama potentially promote monodominance. It was hypothesized that highly abundant EM fungi competed with the saprophytic fungi for nitrogen, slowing down the nitrogen litter decomposition rates. Since these lower decomposition rates result in a reduction of availability of inorganic nitrogen in the soil, the proposed result was increased survivorship and growth rate of Oreomunia seedlings. This in turn would lead to a higher abundance of Oreomunia trees. Let's see how this hypothesis was investigated and tested.
They tested the hypothesis using 3 experiments. For the first experiment, the researchers tested if EM fungi improved seedling growth. Oreomunia and Rupala seedlings were placed inside and outside an Oreomunia forest, and 3 treatments were placed on the seedlings. For the first treatment, a mesh was placed around the seedlings to prevent contact with EM networks. The second treatment excluded root contact with surrounding plants, and the third transplanted seedlings to different soils. Their control group left the seedlings untouched. The seedlings were left to grow for up to 11 months, and the relative growth and mortality rates were calculated for each seedling.
So did the EM fungi networks improve the growth of the Oreomunia seedlings? Using the first 2 methods, they found there were no significant effects of hyphae or the root exposure treatments on seedling growth. However, they did find that the mortality rate of seedlings inside the monodominant forest was significantly less than outside, and the growth rate of the comparison tree Rupala was higher outside of the monodominant forest compared to inside. But there was no difference in the EM fungi colonization of Oreomunia seedlings among the 3 treatments.
For the second experiment, the researchers were testing soil composition effects. Oreomunia trees, as well as 4 other species of trees, were grown in a greenhouse under 2 different conditions. For the first treatment, the seedlings were grown conspecific in their own soil. For the second treatment, the seedlings were left to grow heterospecific in soil that contained the soil of the other 4 species. The seedlings grew for up to 6 months, and the plant-soil feedback, which are plant-caused changes in soil that affect plant performance, was analyzed.
Let's examine the results from the plant-soil feedback experiment. Recall that they predicted that Oreomunia would show stronger positive plant-soil feedback relative to other nearby species. However, Oreomunia showed the strongest negative plant-soil feedback of the 5 species examined.
For the final experiment, researchers were testing how the trees affected the nitrogen cycle. Resin bags were buried beneath the soil surface of the trees inside and outside the monodominant forest for 18 days and then collected. Nitrate, ammonium, and phosphate were extracted from the resin bags and then compared.
They found lower amounts of ammonium and nitrates inside the monodominant forest, but no differences from site to site. Inside the forest, there were higher concentrations of total carbon and nitrogen, and a higher carbon to nitrogen ratio.
The objective of the study was to test the major hypotheses for monodominance in tropical forests, specifically EM networks and plant-soil feedback. The results suggest that neither plant-soil feedback nor the formation of EM networks can account for monodominance. What they did show is that an Aureomunia-dominated forest has lower availability of soil ammonium and nitrate compared to nearby forests without Aureomunia. Thus, EM fungi confer a competitive advantage to Aureomunia seedlings by depleting accessible nitrogen from the soil. In conclusion, Aureomunia monodominance is likely caused by changes in nitrogen availability from EM fungi associations. Reduction in nitrogen availability may explain other monodominant forests. The symbiotic relationship between fungi and plants has existed since the transition to land and plays an important role in the success of both. It is also interesting to understand how biotic and abiotic factors interact to form various ecosystems.
From the Yellow Yak Private Investigation Agency, we would like to say, case closed.